السلام عليكم أعزائي الطلاب. This is the fourth lecture regarding amino acid metabolism. We discussed in the previous three lectures the catabolism of amino acid, whether the amino group or the carbon skeleton. In this lecture, we'll try to discuss the synthesis of amino acid and the specialized product of amino acid. These are the anabolic pathway. First, the synthesis of essential amino, non-essential amino acid, and then the synthesis of specialized product from important amino acids, which are glycine, serine, methionine, histidine, arginine, tryptophan, tyrosine, and glutamate. First, we start by biosynthesis of non-essential amino acids. What are these? Some amino acids can be synthesized from precursors in the citric acid cycle and the glycolytic pathway, such as glyceraldehyde 3-phosphate, pyruvate, and in citric acid cycle such as alpha-ketoglutarate and oxaloacetate. In addition, several of amino acids can be formed from other amino acids. There will be interconversion between amino acids. These two types or two groups of amino acids are considered non-essential because we can synthesize these amino acids inside our body. The other type or other group of amino acids are called essential because they cannot be synthesized inside the body and should be provided in the diet. This is a table represents the essential and non-essential amino acid. We have 10 essential amino acids. They are as the following. It is easy to remember them as the following: 3 basic amino acids, arginine, histidine, and lysine. 3 branched amino acids, valine, isoleucine, and leucine. 2 amino acids starting with the letter T, threonine and tryptophan. 1 aromatic amino acid containing benzene ring, phenylalanine, the precursor of tyrosine, and 1 sulfur containing amino acid, methionine, which is a precursor for synthesis of cysteine. The non-essential amino acids also are 10, but they are here 12 because we included hydroxyproline, which is derived from proline, and hydroxylysine, which is derived from lysine. We will discuss in the following slides the synthesis of these 10 non-essential amino acids. Starting from alanine. Alanine can be synthesized from pyruvate by transamination. By the action of which enzyme? Yes, that's true. It is ALT. Pyruvate plus glutamate would be converted into alanine and alpha-ketoglutarate. We are familiar with this reaction. This is ALT reaction. Aspartate can be formed by AST from oxaloacetate. Oxaloacetate and glutamate by transamination by SST result in the formation of aspartate and alpha-ketoglutarate. We discussed the importance of this reaction in the liver and urea cycle, as aspartate is a provider of the second amino group or amide group in urea. Also, transamination can be served as a source for synthesis of a glutamate. A glutamate indeed can be synthesized in several ways. First, by the reverse of AST or ALT reaction. The glutamate by transamination can be synthesized. Also, glutamate can be synthesized from alpha-ketoglutarate by the reverse of oxidative deamination carried out by glutamate dehydrogenase and the synthetic pathway that requires an ADPH rather than NADH as a coenzyme. The last way is that glutamate can be synthesized by the action of glutaminase on a glutamine to move the other amino group from glutamine and from glutamate. Aspartate and the glutamate, as we said before, can be synthesized by the action of asparaginase and the glutaminase on asparagine and the glutamine, especially in the liver. The importance of asparagine is that it can be used as a synthetic form as an anti-cancer drug, and this subject is discussed briefly in Living Code page 262. Please read it and you will understand. A glutamine synthesis is by glutamine synthetase. This enzyme can utilize the free ammonia, glutamate plus ATP, and the free ammonia by the enzyme glutamine synthetase result in the formation of glutamine, and this is one of the ways that we deal with the free ammonia inside tissues. On the other hand, asparagine cannot utilize free ammonia in its synthesis. Asparagine synthetase utilize the amino group from glutamine. So aspartate plus glutamine and ATP result in the formation of asparagine. And asparagine synthesis is not a way to deal with the free ammonia. Now we have these three amino acids: proline, serine, and glycine. Proline can be synthesized from glutamate by cyclization. We'll see it in the following slide. Serine and glycine are interconvertible by the aid of tetrahydrofolic acid, a single carbon group carrier. Also, serine can be synthesized from precursor of glycolytic pathway from glyceraldehyde 3-phosphate and the 3-phosphoglyceric acid. This diagram shows the synthesis of a proline. We see this is glutamate. Yes, this is glutamate, this is the carboxyl group, and this is the amino group. The carboxyl group and amino group undergo by series of reactions. You don't have to remember these. Cyclization to form the amino group and this ring. So glutamate is converted into proline. And the proline, of course, can be used for synthesis of hydroxyproline. Serine and glycine, as I said, are interconvertible. Serine can be converted into glycine when it loses its methylene group, and tetrahydrofolic acid is converted into methylene tetrahydrofolic acid. On the reverse reaction, glycine can be used for synthesis of serine. Glycine plus tetrahydrofolic acid methylene result in the formation of serine and release of the free tetrahydrofolic acid. Now we came into cysteine. Cysteine is a sulfur-containing amino acid. It is derived from methionine by series of reactions. It is better to discuss it here in this diagram. To synthesize cysteine, we need serine, which is an important amino acid, and we need L-homocysteine. From where we derive this L-homocysteine? Methionine first, this is not shown here, react with adenosine triphosphate with ATP to form S-adenosyl methionine. S-adenosyl methionine is a methyl donor used for transmethylation in several reactions. When it undergoes transmethylation, when it is involved in transmethylation, S-adenosyl methionine is converted into S-adenosyl homocysteine, and then it loses its adenosine group to form homocysteine. So this homocysteine is derived from methionine. You all to remember this. Homocysteine is derived from methionine. L-homocysteine react or combine with serine, forming cystathionine. And here the sulfur group is transported from L-homocysteine into serine, result in the formation of cysteine and L-homoserine. L-homoserine this then divided into alpha-ketobutyrate and a few ammonia. This ammonia group is removed. The remaining is alpha-ketobutyrate, and this free ammonia is dealt with by several pathways. The L-cysteine, so L-cysteine have its carbon skeleton derived from serine. This is the synthesis of cysteine. Tyrosine is synthesized from phenylalanine by an important enzyme called phenylalanine hydroxylase, and this enzyme requires a coenzyme called tetrahydropterin. This is the diagram showing this reaction. Tyrosine derived from phenylalanine by hydroxylation at this position by the enzyme called phenylalanine hydroxylase, and this enzyme requires a coenzyme called tetrahydropterin. You should remember these two, phenylalanine hydroxylase and tetrahydropterin. We'll speak about them on the next subject, which is the inborn error of metabolism. As we said, tyrosine and cysteine are derived from essential amino acid. Tyrosine from phenylalanine, cysteine from methionine. So if we have deficiency of phenylalanine or methionine, if they are not provided adequately in the diet, there will be deficiency of tyrosine and cysteine, and especially tyrosine is important precursor for several specialized products. All of them will be affected if there is phenylalanine deficiency in the diet. We completed the synthesis of non-essential amino acid. Now we'll speak about conversion of amino acid into specialized product. The first of which is glycine. Glycine is a precursor for several important compounds. The first of which is heme. Heme, the essential compound in hemoglobin, requires glycine for its synthesis. Also, it's important for synthesis of purine nucleotide. So it is essential for DNA synthesis. For synthesis of creatine, as we said before, by combination with arginine. For formation of glycine conjugates, example glycocholic acid, derived from reaction of glycine or combination of glycine with cholic acid. And the last is for glutathione synthesis. Serine, used for synthesis of important compound. Phosphatidylserine, an important phosphoglyceride present in the cell membrane. It has a very important function. We discussed it in the previous year. Also, serine important for synthesis of sphingosine, so it is important for phospholipid and sphingolipid. Serine also involved in the synthesis of both purine and pyrimidine in nucleotides. Third amino acid is methionine. Methionine, we said before, is the precursor for S-adenosyl methionine, which is a methyl group donor in various synthetic reactions. Any synthetic reaction that required methylation usually utilizes S-adenosyl methionine. S-adenosyl methionine serves a precursor for synthesis of spermine and spermidine, very important growth factors, and help affect histidine. Histidine, important basic amino acid. By decarboxylation, it is converted into histamine, an important reactive substance, important for allergy, and also considered as a neurotransmitter. Arginine, important for synthesis of creatine with glycine, for synthesis of spermine and spermidine, the growth factors, and it is a precursor for nitric oxide. Nitric oxide, not nitrous. Nitric oxide is a very important reactive substance formed by this reaction. Nitric oxide synthase will convert arginine into acetylene with the production of nitric oxide, and it requires an ADPH. We don't have a single nitric oxide synthase. In fact, we have 3 types of nitric oxide synthase. They are discussed thoroughly in your textbook. You should be familiar with them. Read them, please. The several types of nitric oxide synthesis result in formation of nitric oxide in several places inside the body with different functions. In the blood vessels, it acts as a vasodilator, acts to relax the smooth muscles of the blood vessels. Also, it inhibits platelet aggregation, so it inhibits formation of thrombus. This is inside the blood vessels. In the brain, nitric oxide is an important neurotransmitter. The third position is in the macrophages. In the macrophages, nitric oxides are involved in what is called respiratory burst, a process by which the macrophages produce reactive oxygen species which act as a bactericidal, that is to say, it kills the bacteria and help in neutralize its harmful effect. So there are different functions of nitric oxide carried out by different nitric oxide syntheses at various positions inside our body. Tryptophan. Tryptophan is a precursor of 2 important compounds. The first is serotonin. Serotonin is a neurotransmitter and it has an important function. It is also considered as a hormone. It is called the hormone of love, sometimes called, or of joy. Tryptophan, by hydroxylation, is converted into 5-hydroxytryptophan, and then by decarboxylation is converted into 5-hydroxytryptamine. This is the serotonin. This diagram shows this reaction. Tryptophan, by hydroxylation, it's converted into 5-hydroxytryptophan. Also, tryptophan hydroxylase requires tetrahydropterine, like phenylalanine hydroxylase. Tetrahydropterine is an important coenzyme. 5-hydroxytryptophan then lost its carboxyl group. This carboxyl group is removed, for we have serotonin, 5-hydroxytryptamine. The other compound derived from tryptophan is melatonin. Melatonin is an important neurotransmitter in the pineal body, inside the brain. Serotonin is converted into an acetyl serotonin and then eventually into melatonin. Tyrosine. Tyrosine is very important amino acid. It have several distinct pathways for synthesis of distinct substances. First, it is the precursor for melanin synthesis, which is the pigment in the epithelial cells of the skin. Is the pigment of the skin, melanin, and its deficiency results in the condition called albinism. Also, tyrosine is a precursor for epinephrine and norepinephrine, very important neurotransmitters produced in the medulla and in the neurons. Thyroid hormone also are derived from tyrosine. So, a lot of useful and important substances derived from tyrosine. Lastly, we'll speak about glutamate. Glutamate is the precursor for gamma amino butyrate or gamma amino butyric acid, an important neurotransmitter. Glutamate by glutamate decarboxylase loses its carboxyl group and it is converted into gamma amino butyrate. Also, glutathione, important substance used by the defense system of the body to neutralize reactive oxygen species and reactive nitrogen species and any radicals, is formed from the combination of glutamate, cysteine, and glycine. Here, glycine, we said before, glycine, one of the substances involved in the synthesis of glutathione.